Spermatogenesis, an overview. Spermatogenesis is the process through which male germ cells, or spermatogonia, develop into mature sperm cells, spermatozoa. It occurs in the seminiferous tubules of the testes and involves a series of highly regulated steps, including mitosis, meiosis, and spermiogenesis. The process ensures that sperm cells have half the number of chromosomes, haploid, making them capable of fertilizing an egg. Phases of spermatogenesis. Spermatogenesis can be broken down into three key stages. Spermatocytogenesis. This is the first phase involving the mitotic division of spermatogonial stem cells, spermatogonia, into primary spermatocytes. Spermatogonia. The diploid stem cells that undergo mitosis to produce more spermatogonia or differentiate into primary spermatocytes. These cells are located at the basal membrane of the seminiferous tubules. Type A, spermatogonia. These divide mitotically to maintain the population of stem cells. Type B, spermatogonia. These cells differentiate and enter meiosis to become primary spermatocytes. Meiosis is the reduction division that reduces the chromosome number from diploid to haploid. This phase involves two sequential divisions. Primary spermatocytes, 2N. These cells are formed from type B spermatogonia. Each primary spermatocyte undergoes meiosis I, which is a reduction division that results in two secondary spermatocytes, each with a haploid N number of chromosomes. Meiosis I. The primary spermatocyte undergoes homologous recombination and chromosome separation. This division results in two secondary spermatocytes, each with half the number of chromosomes, haploid. Secondary spermatocytes, N. These are haploid cells that proceed to meiosis II without further replication of DNA. Meiosis II is similar to mitosis, where each secondary spermatocyte divides into two spermatids. Spermatids N. These are the result of meiosis II, with each spermatid containing a single set of chromosomes, making them haploid. Spermiogenesis. Spermiogenesis is the final phase, where spermatids undergo morphological changes to become mature sperm cells, spermatozoa. Formation of the acrosome. The acrosome, a cap-like structure containing enzymes necessary for fertilization, forms over the nucleus of the spermatid. Development of the tail. The spermatid forms a flagellum tail that will aid in motility. Condensation of the nucleus. The nuclear material condenses and elongates, creating the head of the sperm. Cytoplasm. Elongation. Excess cytoplasm is eliminated, leaving the spermatozoon with a streamlined shape. Mature spermatozoa. The final structure of the sperm consists of the head, containing the nucleus and acrosome, midpiece containing mitochondria for energy, and tail, flagellum for motility. Key structures involved in spermatogenesis. Seminiferous tubules. These coiled tubes in the testes are the site of spermatogenesis. The seminiferous tubules are lined with Sertoli cells that provide nourishment and structural support for the developing sperm. Sertoli cells, often called nurse cells. Sertoli cells support and regulate the development of sperm cells. They form the blood-testis barrier and provide essential factors for the growth and differentiation of spermatocytes. Leydig cells. Located in the interstitial space between seminiferous tubules, Leydig cells produce testosterone, a key hormone that regulates spermatogenesis. Hormonal regulation of spermatogenesis. Spermatogenesis is tightly regulated by a feedback mechanism involving several hormones. Gonadotropin-releasing hormone, GnRH. Secreted by the hypothalamus, GnRH stimulates the anterior pituitary to release two key gonadotropins, luteinizing hormone, LH, and follicle-stimulating hormone, FSH. Luteinizing hormone, LH stimulates the Leydig cells in the testes to produce testosterone, which is crucial for the initiation and maintenance of spermatogenesis. Follicle-stimulating hormone, FSH. FSH acts on the Sertoli cells to promote the process of spermatogenesis. It also enhances the action of testosterone by stimulating Sertoli cells to produce androgen-binding protein, ABP, which concentrates testosterone in the seminiferous tubules. Testosterone. Testosterone is essential for the differentiation of spermatogonia into primary spermatocytes. and supports the entire process of spermatogenesis, including spermiogenesis. Inhibin, secreted by the Sertoli cells, inhibin inhibits the release of FSH from the pituitary, providing negative feedback to help regulate spermatogenesis. Factors affecting spermatogenesis. Spermatogenesis can be affected by several factors, including age. Male fertility typically peaks in the late teens and early 20s. As men age, sperm production and quality decline, although it can continue into old age. Temperature. Spermatogenesis is temperature sensitive. The testes are located outside the body in the scrotum to maintain a temperature slightly lower than body temperature, which is optimal for sperm production. High temperatures, e.g. from tight clothing, hot tubs, or fever, can impair spermatogenesis. Hormonal imbalance. Disorders in hormones, particularly testosterone, can disrupt spermatogenesis. Conditions like hypogonadism, low testosterone, or Klinefelter syndrome, extra X chromosome, can lead to infertility. Toxins and environmental factors. Exposure to environmental toxins such as pesticides, heavy metals, or radiation can impair sperm production and function. Lifestyle factors. Smoking, alcohol, and drug use can negatively impact spermatogenesis. Stress and poor diet may also reduce sperm quality. Summary of spermatogenesis process. Spermatocytogenesis, spermatogonia, stem cells divide and differentiate into primary spermatocytes. Meiosis. Primary spermatocytes undergo meiosis to form secondary spermatocytes, which then divide to form spermatids. Spermiogenesis. Spermatids undergo morphological changes to become mature sperm. The entire process of spermatogenesis takes approximately 64 to 72 days from the initial division of spermatogonia to the formation of mature sperm. Spermatogenesis occurs continuously throughout a male's life, although the rate of production may decline with age. Spermatogenesis is a complex but essential process for male fertility, and any disruption in this sequence of events can lead to infertility. Understanding the stages of spermatogenesis and the factors that regulate it is critical for diagnosing and treating male infertility.